will discuss about what is the structure of an antibody. So what are the different classes of antibodies? So how they actually function. Okay. So first we will discuss about the structure. So Potter and Edelman actually they first identified the structure of an antibody, and they were awarded with Nobel Prize, okay, for their work. And they developed these antibodies by injecting myeloma proteins. So myeloma is a type of cancer, okay, skin cancer. So when they induced the skin cancer, usually the body will develop the antibodies. So those antibodies they have studied for the determination of their structure. So antibodies are otherwise called as immunoglobulins. Okay, so they are glycoproteins. That means they are made up of carbohydrate and proteins together. It's a conjugation. So they have few sites of attachment for carbohydrate. So where there are variable regions and constant regions. Within the antibody, you have two regions. One is variable region and the other is constant region. So just remember these terms. Variable means which will be keep on changing. Constant means it remains constant. Okay. So this constant region is made up of carbohydrate. Okay. So first I will give you all this information. Then I will show the structure and I will explain. So meanwhile, you have to remember the terms. Okay. So and the antibody is Y-shaped and its surface is complementary to the antigen. So that is the epitope region. And this Y region, it has two antigen binding sites. Hence, it is called as bivalent region. Okay. And each antibody molecule is made up of 4 polypeptide chains. And out of 4, 2 are identical short chains, which are called light chains. And 2 are identical long strands, which are called heavy chains. So hence we usually write it as H2L2. That is 2 heavy chains and 2 light chains. Okay. And this heavy chain, one heavy chain and one light chain are held together by disulfide bonds. So they are called as heterodimers because in each strand, usually one light chain and one heavy chain were present. And both light and heavy chains have constant region at the C terminal and variable region at the N terminal. Okay, so V region or variable region is at the tip of the Y arm, and this constant region is present in the, which is present in the light chain is called Cl, whereas which is present on the heavy chain is called CH. In the same way, variable region which is present on the light chain is VL and VH is the variable region in the heavy chain. So here the hypervariable regions present in the light and heavy chains are called as complementary determining regions or hotspots, which are also called CDRs. Okay. Now I will explain you using the structure. So here you can see this pink one. So now it is Y-shaped structure. Is it clear? Now the pink one and the red one. Sorry, and red or dark pink color. Okay. The two pink shades, they are light chains. 2 light chains. Now the blue color ones are heavy chains. Is it clear? 2 light chains and 2 heavy chains are present. Now, so if you see the left hand side one, so the light pink shade, that is light chain, and blue one, heavy chain. So those two are held with a bridge, that is SS, indicated SS, okay, yellow color. So that is disulfide. So it's in the same way here also, on the right hand side also, you have a bridge or connection between the heavy chain and the light chain. And in between these two heavy chains also, you can see disulfide linkage. Is it clear? Then you have at the top bits, small portions are called variable regions. So light pink, so both the pink shades, they have VL. As they are the light chains, they have variable regions in the light chains, hence they are called as VL. Then CL indicates constant region of the light chains. Then VH is variable region of the heavy chain. Then in the heavy chain, you have three constant regions: CH1, CH3, CH2, and CH3. Okay. So these are three constant regions. Now the top dimer FAB region, which is called as FAB region. Both the light and heavy chain, the conjugation is nothing but the FAB, which is antigen binding. AB means antigen binding. And the below portion is FC, that means constant. Now you have to remember that at the topmost region is called N-terminus because amino group is present, NH3 plus is present, so that is N-terminus. And this end towards the bottom, it is COO minus, which is C-terminus. Okay. And now each antibody, this complete structure is one antibody. Now one antibody is having two antigen binding sites. So here, if you see the top variable regions, VL, VH, that combination, one combination can bind to one antigen and the other VH, VL can bind to another antigen. Okay. And below, that is called hinge region. Okay. So it's a conjugation of heavy chains. So that can be cleaved by peptide enzyme. Now I will tell you why it's called variable region, because this region has to bind to the antigen. And all antigens, they don't have the same structure, same epitope, right? So, but antibody remains same. Antibody can be either IgM, IgG, IgA, IgD, IgE. Any of these five antibody only has to interact. But the antigen which is coming, that can be anything. It can be a virus, bacteria. Even in the bacteria, you know how many species are there. Then even in a virus, fungus, every day new viruses are coming. So new organisms, then pollens. So any antigen can enter into the body. So this antibody has to alter according to the antigen which is entering. So this antibody should not be rigid. Okay, it should be flexible. Only then it can alter itself and it can interact with the antigen. So that's why it will be variable region. It will be constantly changing. Its genetic component itself, it will be keep on changing. It will be keep on changing its amino acids so that it can interact with the antigen. Got it? So the immunoglobulin consists of two functional fragments, that is FAB fragment, which is antigen binding fragment, that is VH, CH1, okay, and VL, CL1. Now FC portion is the bottom one, okay, and the antigen specificity of the molecule is mainly through FAB region, and the hinge region binds between these two fragments, and hinge region has more amount of proline amino acids and cysteine residues, because cysteine consists of sulfur group. So for binding of disulfide bond, cysteine molecules will be more. Okay. So interchain disulfide bonds will be present. So is it clear with this structure, or do you have any doubts, or should I explain once again? Please respond so that I will continue with the next.
No, ma'am. Clear, ma'am.
Sorry?
The topic is clear, ma'am.
Clear, no? Okay.
Yes.
Okay, fine. So this is, you have to remember the structure. So this is very important topic, antibodies. So first thing is you have to remember the structure itself. So if you know the structure, then you can if you like you have classification of antibodies. So that is mainly based on this structure only. If you know the basic structure, you can easily write the differences between different antibodies. Okay. Now, deduction of antibody structure. So here the same thing I will just explain you like how this antibody can be digested. Okay, like once the antibody completes its function, whatever the cell or antibody or anything, it will be dismantled again and again. New antibodies will be synthesized. Nothing comes from outside. So mostly our body will recycle itself, right? So how this happens is first this is the top one is the antibody. This when digested by peptide enzyme, so the hinge region will be digested first. So in the above structure only, it was mentioned clearly. It is peptide digestible region. Okay. Now the FAB regions, two FAB regions and one FC portion will be separated. Got it? Then,
Ma'am, use the marker, please.
okay. So can you see here?
Yes,
So
ma'am.
here, when digested by peptide, so sorry, you are getting here. See, two FAB regions and one FC region. Are you getting?
Yes, ma'am.
When antibody is digested. If it is digested by the pepsin, okay, FC fragments will be separated, but the top one, so this top portion remains intact. Okay, that means you are getting only one FC, one FAB dimer. Okay, it is intact. Then, sorry. Here, if you see, when it is digested by the mercaptoethanol, so here all the heavy chains get separated and light chains get separated, irrespective of whether it is FAB portion or FC portion or whatever it is. Okay, so two heavy chains and two light chains. You are getting all of them separate. Is it clear?
Yes, ma'am. So FAB portion will remain intact and FC portion will deduct, no?
No, with the mercaptoethanol, heavy chains get separated. It is not now. It is no FAB FC. So only two heavy chains and two light chains are there, no, in the antibody.
Yes, ma'am.
So I will repeat once again. Now in the antibody structure, you have two heavy chains, two light chains. That is clear, no? That
Yes,
blue
ma'am.
color ones are heavy chains, ash color ones are light chains. Now, when they are binding with disulfide bonds, now the top one are FAB region, one ash color and one blue color, and bottom two blue lines, they are FC portion. Is it clear till here?
Yes, ma'am.
Okay. Now, first is peptide digestion. When this antibody is digested by peptide, now only the intermolecular disulfide bond, okay, that is between these two FC and FAB region, that bond will be broken, but the dimer will be intact. Okay, you can see in this image. FAB remains same. 2 FAB remains same. And FC remains same. But they are all separated. They are not associated with each other now. Is it clear?
Okay.
So after digestion with the peptide, you are getting two separate FAB regions and one FC region. 3 strands you will get. Okay.
Okay,
Then
ma'am.
here top one is pepsin digestion. When it is digested by pepsin enzyme, FC remains same. Okay. The bottom FC will get separated and you are getting separate FC chain. But this top one, the top FAB, now in the previous digestion, both of them got separated, right? But that separation is not happening here. Both FAB remains together like Y shape only. V shape. Now it is V shape. But the bottom region, FC portion is getting separated. Only that cleavage is happening. Is it clear?
Yes, ma'am.
Okay. Now the third digestion is mercaptoethanol, which is a chemical. So using mercaptoethanol, there is no FAB or FC, nothing. Now in the antibody, you have 2 light chains and 2 heavy chains, right? So all of them get separated. You will get 2 heavy chains separately, 2 light chains separately. Only when they interact, you are getting FAB, FC regions. When everything is separated, there is no antibody structure itself. Is it clear?
Yes, ma'am.
At the bottom, 2 blue lines, 2 ash. Completely the antibody is getting dismantled. So this is mainly this kind of digestion. It is used in the laboratory. Within the body there is no mercaptoethanol and all these things. So only peptide and pepsin they will be digesting. Okay, but chemical digestion and all this happens in the lab for the research purpose. If they want to know the structure, if they want to explain the genetic composition of the chains or amino acid sequence like that. Okay. Now,
Okay.
what are the types of immunoglobulins? So, there are 5 major types of immunoglobulins: IgG, IgA, IgM, IgE, IgD. So, all of them they differ in their size, charge, amino acid sequence, and carbohydrate content. And again, within the IgG also, there are different groups like IgG1, IgG2, IgG3, IgG4. They are subclasses. Okay, and their function also will differ. So now first we will discuss about IgG. So IgG is generally found in most of the cases. So it's abundant antibody. So most of them, like in the I told you in the yesterday class, I think. So secondary reactions usually they produce IgG. Okay, so they are present in the serum. 80% of the total serum immunoglobulin is IgG. Okay, it has molecular weight of 146 kilodalton and its half-life is 23 days approximately. So 23 half-life means how much time they can survive within the serum, half of the molecules. So 23 days they can actually survive. So the major immunoglobulin during secondary immune response, right? So whenever an antigen is encountered for the second time, and it can be transported through placenta, and it has 4 subclasses: G1, G2, G3, and G4. They all differ according to molecular weight, interchain disulfide bonds, and functions also. So here are the images of G1, G2, G3, G4. So no need to remember that much depth. Only if you get an essay answer, so you have to write in detail. Okay, you just read it once, you study, but you just remember the general points of G1, GM, like that. Okay, general classification. But it's okay if you, it's good that if you can remember this also. So one serum protein, which is called C1q, it binds to the CH2 domain on the IgG. And this, so CH2 I told na, constant region. So which is present in the FC region. Okay. So it binds and it activates the serum protein, which are called complements. Okay, that pathway will get activated which is a part of the immune response. So innate immune response. So innate is the one which comes through our birth. Okay. And many cells which are including polymorphs, monocytes, macrophages, B cells, NK cells, natural killer cells, T cells, all of them have their IgG at their surface through Fc region, or we can also call as Fc receptors. Okay, here is the below table which explains the difference between these four groups. So which is the abundant one? IgG1 is the most predominant one, which is having 66% in the serum. Serum half-life is also all the three, G1, G2, G4 are having almost same half-life except G3 which is having less half-life. The number of interchain disulfide bonds are more in IgG3 compared to other three. So all of them can cross placenta, okay, but complement activation is lacking with respect to G4. Okay, so just remember this kind of differences. Next is IgM. IgM around 10% it constitutes. Its molecular weight is 970 kilodalton. Its half-life is around 5 days. So it's a first class of antibody made by B cell. Okay, and it is involved in the peripheral circulation, and it is mainly involved in primary response. Immediately when an antigen enters into the body, this IgM will be produced. It is the first antigen to be synthesized in a neonat, that is babies. So it comprises 5 or 4 chains. So here you can see it's a pentamer usually, not a single Y shape, but 5 antibodies with together pentameric molecule. And the heavy chains have 4 constant regions, and the whole unit is actually, it is like joined by a J kind of chain. Okay. So IgM and IgD are found in the B cell majorly. Okay. Then next is IgA. So IgA is around 15 to 20% of the circulating immunoglobulins, and it is mostly in man, it is monomeric, but sometimes it can form dimer, that is 2 chains can combine. So whenever the 2 chains are combined, it is combined by held together by a J chain. So its half-life is 5 days. Then this dimeric IgA, it passes through the endothelial cells, which are usually present in the blood vessels and all these tissues. It acquires secretory components. So usually IgA is a secretory form, and it has strong covalent bond, and it helps in the release through the mucosal surface. Okay, so during this process, so it has to get protected from the proteolytic attack. Other proteins will be present, which will be digesting. So that protection is acquired by this covalent bond. And this IgA is more abundant in the secretions like saliva, breast milk, tears, mucus of bronchia, then genitourinary tract, digestive tract, etc. So here also different classes, IgA1, A2. A1 is the predominant in serum, A2 is predominant in mucus secretion. And in the colostrum of the milk, so IgA usually passes to the babies. Next, IgE. It is present in very trace amounts, like less than 1%. Half-life is only 2 days, and mainly present in the surface of the mast cells and basophils. And it is produced in response to allergic reactions, and it activates the classical pathway of the alternate pathway. So this complements, I told you, serum proteins. So these complements can be synthesized by 2 pathways. So which is classical pathway and alternate pathway. So here IgE activates alternate pathway. Okay. And even IgE can also protect the body from the helminthic infections like parasites. And it also mediates hypersensitivity reactions, uh, which is responsible for the hay fever, asthma, hives, anaphylactic shock. Like to the pollens and allergic reactions. Okay. Then IgD, this is also very trace present in a very trace amount, like less than 1%. Half life is only 2 days, and IgD along with IgM is present on B cells, and it is not much effective. It is not showing any effector functions. Okay, very rarely towards some allergic functions, allergic reactions, this IgD can be produced. This is the classification. So
Ma'am,
here,
in IgA,
hmm,
that figure is of IgE. Then IgA is
here the first one,
first.
immunoglobulin A, blue color one.
Okay.
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 Yeah. So this is the table. So this is good if you can go through. So whatever the points I have told. So even in the table also, only you can just remember. And one more thing is like this heavy chains. I told you, no, heavy chains. So they are also different types: mu, gamma, delta, alpha, epsilon. So first one is mu, IgG is gamma, IgA is alpha, IgE is epsilon, and IgD is delta. So they are nothing but the genetic variation. Okay, so that you have to remember. Then number of antigen binding sites you have to remember, because IgM is pentamer, it will have each antibody 2 antigen binding sites. So 5 into 2, 10. Like that you have to remember. So molecular weight, it is not much necessary. Okay, you can remember. Obviously, if it is a pentamer, it will be heavier. So that is you can just exclude. Then percentage, how predominant it is. So that is important. Whether it crosses placenta, whether it fixes complement. Then this function and Fc binding site also. So that portions you have to remember. Okay. Then functions of immunoglobulins in general. So first one is neutralization. So neutralizing antibody, it blocks parts of the bacterial cell. Okay. So neutralization means it will stop, it won't kill the antigen. It will only neutralize. That means it will stop its spread. It will stop the infection. So they will bind to the surface of the bacterial cell and makes it ineffective. So this neutralizing antibody, it defends a cell from the pathogen or infective particle. Okay. So that it will make that foreign material no longer infectious or pathogen. Okay. So this is a part of humoral or adaptive immune response. So what is humoral? Humor means body fluid. Okay. So humor means the immunity, humoral response is the immune response which is mediated by the body fluids like serum, plasma, saliva. So what are present in this serum, plasma, saliva? Okay. So there might be antibodies, okay, secretory antibodies and cytokines, chemokines. So any secretory material which are present, so that will be actually interacting with the antigen. So it's an adaptive immunity. So it is not the innate immunity, it's a part of adaptive immunity. And it is very specific in nature. The next is agglutination and precipitation. So these antibodies, when combined with the surface of the microorganism or antigen, so this antigen antibody reaction, it produces agglutination or precipitation. So if you in laboratory, if you perform practicals, so where already you will, or you can see the blood grouping. I think you have you done blood grouping?
Yes, ma'am.
So you will see the precipitation, right?
Yes, ma'am.
Like in the blood which is positive, either it is A, B, or O.
It shows clotting.
Yeah, clotting. It's nothing but the agglutination. Precipitate, small precipitate like thing will form. So even in that is also a type of antigen antibody interaction. So if you can see any other antigen antibody interactions also, some kind of precipitation takes place. If precipitation is there, then we can say it is a positive response. Okay. So that is another function. Then complement activation or fixation. So whenever antibodies they interact with antigen, they will activate complement cascade. So here complements are the proteins which are present in the serum. So they will remain inactive until there is an antigen antibody interaction. So once there is an interaction between antigen and antibody, they go and activate the complement system, and it can be through classical pathway or alternate pathway. And these complements, they will activate through a mechanism, and then they will attack the pathogen. Okay. So how they activate? By inducing inflammatory response or by attracting phagocytes. So dendritic cells, all these macrophages, all these are phagocytes. So which will engulf or which will eat the antigen. Then opsonization of cells. Opsonization means it is also like moving towards antigen. So it will produce more response for killing the antigen, and lysis of bacteria or virus. So these are the mechanisms. The next is antibody dependent cell cytotoxicity, which is also called ADCC mechanism, where IgG, it makes the natural killer cells to recognize the target cell, whether it is a cancer cell or anything from foreign. So and they will kill. Like here, other types of cells like monocytes, neutrophils also help the natural killer cells, okay, which will kill the extracellular pathogens. This is a cell mediated. So I told you humoral means it is a body fluid mediated through antibodies or so. Here it is cell mediated mechanism. Then opsonization is nothing but it is a phagocytic mechanism which will eat the microbial antigen. So IgG and IgM antibodies, so they will actually they move to the site of infection, and they will proliferate, like they will induce the proliferation of these phagocytes, and they will help these phagocytes to kill the bacterial or viral particles. Okay. Now we will discuss B cell and T cell activation. So first is B cell activation. So B cell activation requires antigen. Okay. So it can happen to two pathways: TH cell dependent, TH cell independent. TH cell dependent means helper T cell dependent and helper T cell independent. Now here you can see both the pathways are here. Directly I will explain you the figure. So in the T cell independent mechanism, so first the IgM is secreted. This is the first step. Whenever an antigen enters, so this IgM already that will be present in the serum. So this IgM, first antigen, it comes and binds to the surface receptors of the B cell. Okay, directly antigen can come and bind here. There is no need of presentation or anything. Okay, directly this antigen comes and binds to the receptor present on the B cell. This results in the IgM secretion. So because it is a primary response. So this is here no T cell is involved. Okay, and this IgM can trigger, it can like activate the complement pathway and so and so. The mechanism will take place. In the T cell dependent pathway, this antigen goes, it is processed. First, antigen will be processed by the dendritic cells, and it presents this antigen to T cell receptor through MHC class 2 molecule, and this T cell now presents this antigen to B cell. Now this B cell will secrete IgG. Okay. So here whenever a T cell is involved, IgG will be secreted. So whenever in the IgM is secreted, it takes 4 days, within 4 days. Whereas this IgG secretion, it will take more time. Okay. So got it? That is the main difference. And next is T cell activation. How T cells are activated. So they are actually developed in thymus gland and they are matured in the bone marrow. And the T cells, I told you, cytotoxic T cells, helper T cells. And they will be having antigen presenting cells. Directly an antigen cannot go and bind. So antigen presenting cells, they will process this antigen and they will present this antigen using MHC class 1 or class 2 to the T cell receptor. Okay. When they are binding, whenever an antigen comes and binds to this T cell receptor, then cytokines are produced. These cytokines again activate other immune cells and then the killing of the antigen takes place. So this is the T cell activation. So whatever I have covered in the previous classes, like previous topics, so all those like you have to remember. So MSC class 1, class 2, on which cells it is present and all that. Okay. Now antigen antibody interaction as a tool for the research. So I told you antigen antibody interaction is very important and many serological reactions are based on this antigen antibody reactions. So here there are 3 stages. The first stage involves the formation of antigen antibody complex, and in the second stage, once interaction takes place, it can result in precipitation or agglutination. Then in the third stage, this antibody will neutralize or they will destroy the antigen. So based on this, actually, we can confirm whether it is a positive test or negative test. And here there are some properties which are called affinity, avidity, and cross-reactivity. Affinity is nothing but the interaction between an antigen and antibody which are non-covalent. That is between epitope and the paratope. So how many interactions are taking place? It is not covalent one, non-covalent reactions. So if more interactions are there, that is called high affinity. If less interactions are there, it is low affinity. Okay. Then avidity. Avidity is nothing but the strength of the bond. It is not the number of reactions, but it is the strength. Okay. This is the overall capacity of antibody to combine with an antigen. Some antigens will be multivalent. So they have to interact with multiple antibodies. Only then it can be neutralized. So how strongly an antibody can combat this antigen? So that is the avidity. Then cross-reactivity. Here, a particular antibody can react with 2 or 3 antigens. All will have the same epitope. Yesterday, I think I told some kidney cells of humans and some other organism, some bacteria. Okay. So both of them are having same epitope. So that is nothing but the cross reactivity. So same kind of reaction can be expressed. Okay. So where, in which tests can you do, like in AIDS, then rheumatoid arthritis, then VDRL. Okay. So all these pregnancy test. So in all these tests, actually you can detect this antigen antibody reaction. Then monoclonal antibodies. So they are nothing but a single type of antibody, but they have multiple antigenic specific sites. That is nothing but, like if you see IgM, it is pentamer, right? So they are polyclonal, like each antibody can bind to a different antigen. But in the monoclonal, it is a single antibody which is directed to a single antigen. So it is very specific towards the antigen. So usually we can see in the normal body, polyclonal antibodies will be seen. But monoclonal antibodies are specially designed for the research purpose. Like Kohler and Milton, they have developed this monoclonal antibodies, and they were awarded with the Nobel Prize. So they are for the immunization purpose, for the establishment of hybridomas, cell fusion, then for like treatments. So in such cases, usually these are used. I will just explain you in brief. So immunization is nothing but a vaccination. Okay. So the injections of multiple types can be multiple times they can inject. Okay. And they will get a strong immune response. Okay. Then fusion of myeloma cell, which is a cancer cell, along with the B lymphocytes. So this fusion, this is because the myeloma cell is a what you call immortal. The cell cannot die because it's a cancer cell. And B cells will produce the antibodies. So when the fusion takes place, immortal, that is called hybridoma, and that cell become immortal, and it can produce any number of cells, and more amount of antibodies can be produced. And this you cannot see with the naked eye, right? You have to select this more hybridomas from the other myelomas and B cells. That selection is based on the HGPRT enzyme. So that means this is called hypoxanthine guanine phosphoribosal transferase. So this is because B cells contains HPRT1 gene which codes HGPRT. And this HGPRT is involved in the synthesis of nucleotides in the culture medium. Therefore, B cells can grow in the hat media, which is having HGPRT. But this hybridoma, myeloma cells, they cannot grow because they don't have this enzyme and gene. So therefore, even hybridomas, they cannot grow. Okay. So like that, hybridomas can grow, but only myelomas, they cannot grow. So this is again a separate topic, so I'm not going in depth. Okay. So how they'll culture the hybridoma cell and they'll inoculate the hybridoma cell in the host, and they can produce more amount of antibodies. So just remember this, that is hybridoma is nothing but myeloma and B cells. Myeloma is a cancer cell, so it is immortal. It can divide rapidly. Whereas B cells, they can produce antibodies. So when the fusion takes place, the hybridoma cell has the capacity for immortal, can divide number of times, and they can produce more amount of antibodies. And the applications of monoclonal antibodies is mainly in the tumor identification, that is detection, diagnosis. That is, they will develop cancer specific monoclonal antibodies. Okay. That is diagnostic testing. Then cancer therapy or immunotoxicology for the treatment, along with the chemotherapy. And autoimmune diseases. So in some autoimmune disorders and like allergic asthmas. So usually this monoclonal antibodies will be given. So to prevent that. Then transplantation for like bone marrow transplantation, liver, kidney, heart, etc. for the suppression of immune response. Then protein purification. So this is also for, suppose we have developed using biotechnology, usually we produce various proteins using recombinant DNA technology. So using that, whatever the protein we are producing, to purify that protein, also we use this monoclonal antibodies. So these are the applications of the monoclonal antibodies. So this antibodies topic is very important. So like classification of these antibodies, it can come even IgM, IgG. They are very important. They can come as a short answer also. But or like as an essay also, you have to explain all the classification. And this monoclonal antibodies, hybridoma technology it can also come as a short one. So this whatever the figure is there, no? So this figure itself, if you can explain this hybridoma technology and monoclonal antibody production, so this itself can come as a short one. Okay. So in the antigen topic, so what are the characteristics or the properties of an antigen? Okay. So these are some of the important topics in from the topics which I have covered till now. So, so actually all these are important topics only, complement system, MHC molecules. But I'll see in the next, we have only 3 classes. So I'll try to cover some topics from the biotechnology part also. Okay. But you have to read everything. If I am leaving means don't just neglect. Okay, even complement pathway is important. MHC molecules, they can come for short also. And even the complement pathway, you have 2 pathways, classical pathway, alternate pathway. So both of them can combine as essay also. We don't know how you will question paper may come. So focus, you just have to go through once, right? I will see if any other important topics are from animal biotechnology. I will try to cover. Okay, any doubts in today's class?
No, ma'am.
No, ma'am.
Okay, okay then. Thank you so much.
Thank you.